Hello all. In this video, I'm going to explain about the anther or pollen culture. And coming to the introduction of anther or pollen culture, here the anther culture means culturing of anthers in vitro conditions on a suitable nutrient medium is called as anther or pollen culture. Here the microspores which are present within the cultured anthers, these are developed into callus or embryoids, and these embryoids can give rise to haploid plantlets either through the organogenesis or through the embryogenesis. And mainly the basic principle of the anther culture or pollen culture is the production of haploid plants. And generally the culturing technique is based on the totipotency of microspores and the occurrence of single set of chromosomes in the microspores. And coming to the history of anther culture, the anther culture was first carried out by a Japanese cytologist Shimakura in the year 1934 with the aim of understanding the physiology of meiosis. Later, the scientists Guha and Maheshwari accidentally noticed the development of embryo-like structures inside the cultured anthers. Subsequently, the scientists Borgin and Nist obtained the first haploid plant from the isolated anthers of Nicotiana, the Bacchum plant. And during the last decade, much progress has been made in the anther culture of rice, wheat, maize, potato, belladonna, and other crop species. In this picture, you can clearly see the what is anther and what is pollen. Here, this is the flower. Okay, flower contains the sepals, petals, androecium and the gynoecium. Here you can see here the anthers, anther lobes, and within the anther you can also see the microspores. These dark yellow color parts are the microspores. Okay, here the culturing of this part in vitro conditions on a suitable nutrient medium is called as the culture anther culture. And next, coming to the culture technique for raising haploid plants from the microspores or from the pollen grains. Generally, we have to take the entire anther for culturing process. First, we have to select the mature anthers and excised from the flower buds. Then after excising the mature anthers from the flower buds, we have to do the surface sterilization with using sterilizing agent, that is ethyl alcohol or chlorine water. Then after sterilization, we have to transfer these anthers aseptically on the nutrient medium. And these cultures are then maintained in alternating light and dark periods. Here the light period time duration is about 12 to 18 hours and dark period time duration is 2 to 6 hours at 28 degrees Celsius. And during the incubation period, in responsive anthers, the wall tissues gradually turn into brown color. After 3 to 8 weeks, they burst open and exposing the multicellular tissues. In some species, these tissue gradually organize into globular embryo, and these globular embryo grow into a single haploid plantlet. By this method, only we can get single haploid plant from one pollen grain or from one anther. The example of this type of method you can see in the Atropa belladonna and Datura and Nicotiana plants. Alternatively, the majority of the plants, the tissue divides repeatedly to form callus. From this callus, several embryoids are differentiated. Here, each embryoid grow into a plantlet. These plants obtained from the callus are usually mixoploids due to the endopolyploidy. Okay, this is about the technique of anther culture. Okay, after formation, after exposing the multicellular tissue from the bursted pollen grain in one way, it is develops into one embryo. From this embryo, we can get only one haploid plant. In alternative way, the multicellular tissue gradually differentiated into callus. From the callus, we can get several embryoids. From each embryoid, we can get one haploid plantlet. Okay, this is about the technique of anther culture. Then next we will see cytology of androgenesis. The in vitro development of haploid plants obtaining from the totipotent pollen grain or totipotent microspore is called as androgenesis. Through detailed cytological investigations, there are 4 pathways have been identified which are leading to the in vitro androgenesis. And these 4 pathways are pathway 1, pathway 2, pathway 3 and pathway 4. Okay, in pathway 1, the microspore or pollen grain divide equally and both daughter cells contribute to the further development of haploid plant or haploid sporophyte. And here there are no vegetative and generative cells of differentiation. The example of pathway 1, Datura inoxia. In Datura inoxia plant, the androgenesis through the pathway 1 and pathway 2. In pathway 2, the microspores divides by an unequal divisions to form vegetative cell and generative cell. Here the haploid plant arises from vegetative cell only, not from the generative cell. Only vegetative cell only contribute for the development of haploid plant. The example of pathway 2 is Nicotiana tabacum. In Nicotiana tabacum plant, microspore divides unequal divisions from the vegetative cell only. The haploid plant of Nicotiana tabacum will arise. Then next, third pathway, that is the pathway 3. In pathway 3, the microspores undergoes unequal divisions like in the pathway 2. But here the haploid plant arises from the generative cell only, unlike in the pathway 2. In the pathway 2, the haploid plant arises from the vegetative cell, and in the pathway 3, the haploid plant arises from the generative cell. The example of pathway 3, Hyoschemus niger. And coming to the last pathway, that is the pathway 4. Here in the pathway 4, the microspore divides unequal divisions like in the pathway 2 and pathway 3. But here, like in pathway 1, both cells, the vegetative cell and generative cell, contribute to the formation of haploid plant. The example of pathway 4, Datura inoxia and Datura metal. Okay, after differentiation of this microspore, here in the second cycle, you can see the bursting of anther wall and exposing the multicellular tissue. And these from these multicellular tissue, in one way we can get one embryoid, and in alternative way we can get callus. In which way only one embryoid will form, from that way we can get only single haploid plant, and in which way we can get callus, from that way we can get more haploid plants during the further culturing process. Okay, next we will see factors which are affecting on the anther culture. Here there are 4 factors are affect the culturing of anthers. They are temperature, culture medium, stage of pollen at the culturing process. 4th effect is the physiological status of donor plant or selection of anthers. First we see temperature, how the temperature will shows their effect on the culturing of anthers. Here high temperature, that is more than 20 degrees Celsius, had a promotory effect on the pollen embryogenesis in the tobacco and datura plants. And in datura plant, the optimum temperature for anther culture is 25 degrees Celsius. And also a cold treatment to the flower buds before excising the anthers, it is increase the output of embryoids. And the scientist Nish reported in the plant Nicotiana tabacum, up to 58% of anthers yielded embryos from the buds which we are kept at 5 degrees Celsius for 72 hours. In contrast, only 21% of anthers yielded embryos if the birds were kept maintained at 22 degrees Celsius for the same period. Means here the scientist Nish proved that is the cold treatment in the case of Nicotiana tabacum will promote the formation of embryos in the 72 hours. And when the Nish kept the Nicotiana tabacum anthers in high temperature, in that case only 21% of anthers yield embryos for the same period, that is 72 hours. The next factor is the culture medium. Here the nutritional requirements of excised anthers are much simple in compared to the other culturing process, because the anthers can grow on the basal medium which are supplemented with the coconut milk or kainate. Of the various mineral salts, here the macronutrient iron plays an important role for the induction of haploid plants. And coming to the carbohydrate supply, here the sucrose is generally used at the concentration of 2%. And the addition of activated charcoal, that is 0.5 to 2% into the culture medium, it is stimulate the androgenesis during the culturing process of anthers. And then next, third effect is the stage of pollen during the culture time. And here the age of the donor plant and the environmental conditions in which the plants grow and influence the androgenesis. Generally, the anthers from young plants are more responsive, and the anthers which are taken from the short day plants shows relatively better response than the long day plant. For anther culture, we should take the anthers from young plants or the anthers from short day plants, because these anthers only responsive anthers for getting haploid plants through the culturing process. The next factor is the physiological status of donor plant or the selection of anthers. The anthers which are having uninucleate pollen grains are the most convenient for the anther culture. For the maximum production of pollen plants, the appropriate stage of microspore development varies with both the plant species. For example, in Datura and tobacco, the anthers give best response if the pollen is cultured at first meiosis stage. And in most other cases, the anthers are most productive when these anthers cultured at uninucleate microspore stage. In the ideal stage in the Lycopersicon plant, it has been found to be the microspore mother cells in the meiosis one. And in another hand, in Brassica species, the anthers with the mature pollens are most suitable for the culturing process. Okay, these are the factors which are effect on the culturing process of anthers. The next is the diploidization of haploids. The haploid plants, after culturing process, when once we get haploid plants, these are shows a normal growth up to flowering stage. But these plants cannot produce viable gametes due to the absence of homologous chromosomes, and the plants remain sterile and there is no seed set. These haploid plants may be perpetuated by the duplicating the chromosome complement. For the duplication of chromosomes, we have to treat that haploid plant with 0.4% of colchicine for 28 to 48 hours. After treating with the colchicine of 0.4%, chromosome duplication will occur in the haploid plants, then it will change into diploid plants. This is about the diploidization of haploids. Then next, last, importance of haploids. Here the homozygous diploid plants can be used as pure lines in the breeding programs. Here the haploid plants are useful in the cytogenetic studies. The recessive mutations can be easily detected in the haploid plants than in the diploid plants because they contain only one set of chromosomes. Haploid plants become a good source of biochemical and for physiological studies like microorganisms. And homozygous diploids are beneficial when the plants are self-incompatible. The example of self-incompatible plant is rye. And also the haploid plants are useful in the genetic engineering. In the genetic engineering, the haploid plants can be successfully used for the gene transfer. And the haploid tissue of Lycopensicum has been used for the transfer and expression of 3 genes from the Escherichia coli. These are the main importance of haploids.